Okay, so this is the interesting thing about antibodies. So antibodies, they are proteins, they are chemicals. So you can use antigens to attack foreign pathogens and antigens, but antibodies can also be used to detect things. And this is the basis behind a lot of lab tests, especially if you're going to med tech or yeah, any sort of like lab work. This was something you'll definitely need to know. So there is a certain type of scientific procedure we call, or scientific method we call ELISA, so enzyme linked immunosorbent assay. So what it does is actually uses antibodies to detect substances. Remember, antibodies are chemicals. They can bind to chemicals. So you don't necessarily need to make an antibody against something that causes disease. You can actually make antibodies against hormones and metabolites. Even things like you can generate anti-testosterone or like even though it's not a protein, it's a chemical. So you're able to act, if it's a chemical, in theory you can generate an antibody against it. So what happens is like pretend this target antigen is like you're measuring the amount of like a hormone in the body. You can use antibodies that bind to that hormone and use a bunch of antibodies in succession. And what this is basically an antibody that generates light. So by having more of this hormone in this case, or antigen in this case, by binding more and more antibodies, you generate more light. So this is kind of a way that you see in a lot of medtech labs that they measure amounts of substances and hormones and other chemicals in the body. So ELISA is a powerful method. But this is what I'm getting at. What's more applicable to this class is blood typing is detection. So when you're doing this, like I know everything's virtual this semester because of like restrictions, but blood typing is kind of like that. So what it does is like antibodies can be used to detect things, and what we have is like you need blood typing to determine what someone's blood type is. So it's more like it's a screening process to make sure that your donate or that you blood type a donor's blood before you actually transfuse it into a patient to maximize or minimize the risk of that having an adverse reaction in that patient. So this is how you do it. You obtain serum with these antibodies, so either anti-A, anti-B, or anti-Rh antibodies, and then you mix with a blood sample from a patient. You don't mix it in the patient because again, you don't want that hemolytic transfusion reaction within that patient. And then you check for agglutination, so you look for that clumping. Alright, so let's go to top hat. Now, agglutination, remember, so things that your blood, and you've seen your own blood, right, at one point, right? Like everyone's had a cut. So you notice that's pretty diffuse, at least without looking at a microscope. It looks uniformly red. But when you have clumping, that's when it starts to form like little, like it's not so a consistent color. So does this person have A antigens on their red blood cells? So look at this. Which looks clumped? Which doesn't look clumped? Which looks like blood that typically when you cut yourself, it looks like a blood stain, and which looks like not something you would hope to see oozing from your blood vessels when you cut yourself. A further hint, this is not agglutinated, this has agglutination. This has agglutination. Alright, let's see how people did. So close this. Alright, most of you said, does this person have A antigens on the red blood cells? Again, look here in the anti-A. So you're mixing antibodies with the patient's red blood cells. Notice there is agglutination. Yes, they do have anti-A, or they do have A antigens. Why? If they didn't have A antigens, there would be nothing for the anti-A antibodies to bind to. So yes, because there is agglutination, they have A antigens. Next question. Does this person have B antigens on their red blood cells? If you need a further hint, Notice that the B reaction is totally different from the A reaction. Alright, let's see how people did. So most of you said no, and most of you are correct. Again, A definitely has a agglutination, and B, notice that it's pretty diffuse, it doesn't have the clumping. Therefore, why? Because you have the anti-B antibody in this sample, but with a lack of antigen, with a lack of B antigen, there's nothing for the B antibodies to grab onto. So yeah, when it says anti-A, that means you have anti-A antibodies in the sample, in this little circle here. With anti-B, you have anti-B antibodies in the circle here. Now for this last one, this is anti-D. And yes, you can see agglutination with your naked eye. So it's pretty obvious. Like, it doesn't look nice and uniformly red or diffuse red. Okay, last question for this sample, or actually second to the last question. Does this person have RH or the antigen D factor on their red blood cells? And yes, as someone pointed out, you would have, in this sample here, you have the the blood of the patient being tested, but you also have anti-RH antibodies in here. Okay, let's see the results. Most of you said yes, and most of you are correct. So again, there is agglutination, there's this clumping, it's not diffuse and uniform like this color here. So yeah, whenever you see this clumping, that means, so if you're using this type of blood typing kit, when you have agglutination, that means that antigen is there. So if you have clumping in the anti-A, that means they have the A antigen. If you have clumping in anti-B, that means the patient has the B antigen. If you have clumping here, that means that they have the RH factor. So again, this is not testing the patient's antibodies, it's testing the red blood cells. It's just that in these cards, you already have the antibodies, so it's really cool. Like, they actually have it freeze-dried or dehydrated, and you just resuspend it in water, and these antibodies can attach to and agglutinate cells. Now let's put this all together. What is this same sample? What is this person's blood type? So for this class, these are the only 8 blood types I'll test you on. Okay, so most of you said A positive, and again, anti-A. That means we do have agglutination, we do have the antigen there. So therefore it is A something. B, there's no agglutination, therefore that rules out B, that rules out also AB as well. And again, the first column also ruled out O. So it's A something. With the agglutination in the anti-D RH, that means we do have the RH factor. Therefore, the people who said A positive, correct. For this question, you get A plus. And so again, this is the person's blood type. Alright, so then blood typing, this is what we have here. So again, if there's agglutination, that means that the antigen is there. So anti-A, you have agglutination. Yes, there is the agglutination. Yes, there is a A antigen. Anti-B, no agglutination, therefore no B antigen. For anti-D, yes, you have agglutination, therefore you have the RH factor. So this is when you have a yes, that means you have the antigen. If there's a no, there is no antigen of that type. And then if there is agglutination, you have that antigen in that same column. This is why it's A positive. So for this one, I'm going to go over another example and let's ask the chat. Do you see, it's a little hard to see in this example, but is there agglutination in the A column? Does it look uniform in color, or does it look kind of spotty? Yep, so you do see agglutination. How about in the B column? Do you see agglutination, or yes or no? Yeah, this one's not, don't worry, this image, like, I'll find better quality images compared to other for the exam, but this is just an example. Yeah, it's kind of hard to see, but notice that you have light areas and then you have these dark red patches as well, so it's not quite uniformly colored, so you do have agglutination there. How about anti-D. So this is actually a shine here in this blood droplet. So do we see a agglutination there? Yep, so no, we don't see a agglutination. So this is how you read these cards very easily. So yes in the anti-A, therefore this patient has the A antigen. Yes to agglutination in the B column, therefore yes, you do have the B antigen. No agglutination in the anti-D/RH column, therefore they are Rh negative. This person's blood type is AB negative. So this is why, again, a lot of times people get confused with blood typing. They think, oh, this is detecting the patient's antibodies. The antibodies are actually on the card itself. You're actually detecting the antigens on the patient being tested. So again, this is how you use that. If you see agglutination, you put a check mark next to that antigen. And that's how you're able to use it. Okay, so we're definitely over time, and again, study guide, I will be working on this right after this class, so it'll be up later on this evening, all right? So Friday will be a review session. So I'll have two ways you can submit questions. You can either submit questions on Top Hat, and I'll go over, sometimes if they're easy, I just answer it on Top Hat. But yeah, Friday is all a Q&A session. No news, nothing new, and we didn't get to cover heart anatomy. So everything cardiac, I'm pretty much saving for Unit 2. Yeah, so there's no heart anatomy on exam 1 because I didn't have time to get to it yet. Yeah, I figure rather than rush through blood typing and squeeze in what heart anatomy I can, just save it for the next exam.